program. And so we thank him very much for that. And I think, shall I give you a little biographic back? We've been at UEA for a very long time.
About 10 years, yeah.
10 years.
That's right, yeah.
Yes. And you were trained as a mathematician?
Yeah, that's right, in America. That's right. Yeah. Sort of moved on from there.
Yes. Good.
Yeah.
Okay, well...
okay, thank you very much, yeah. So today I'm going to talk about some work that we've been doing partly at the University of East Anglia, but I thought I'd start off by actually making a lot of acknowledgements because this is really quite a large project by now. But the key players that I've been working with over the last 3 to 4 years have been Thomas Mock, who's there. He's at University of East Anglia. He came about 4 years ago there, and he was very interested in all the work that we're presenting now. And I started to talk with him, and I thought that sounds like a really exciting project. So maybe that would be fun to get involved with. And we managed to find some funding for a PhD student at that stage, who's Andrew Toseland, who's basically done a huge amount of the work on the sort of metatranscriptomics analysis that I'll present. And Andrew's a bit delayed today. He's going to hopefully come later on, and he's giving a little poster outside. So if you have sort of more technical questions about that, I'm sure he'd be happy to answer those. This work then has sort of grown. I mean, obviously there are a lot of people involved from different institutes who have helped either with the sequencing or collection, so University of Algarve, the Alfred Wegener Institute, but more recently we sort of teamed up with a group at the University of Exeter with Tim Lenton, Stuart Daines and James Clark, and they basically have done all the modelling that I'm going to very briefly present at the end. So this is far from being all my own work, yeah. In fact, it's a very small fraction of it is my work. So there's Andrew's poster anyway, so that's just out in the foyer, and you're welcome to go look at that. So as Wally said, I'm a little bit out of my comfort zone here because I'm trying to talk about a rather big picture, but I thought rather than just to sort of stand up and go through some metagenomics or metatranscriptomic results, I'd try and put it into a bigger picture, and maybe out of that, as the program develops, we'll find interesting connections and possible future directions. So marine phytoplankton are the things that we're mainly interested in here. So those are microorganisms that inhabit the upper sort of sunlit layer of the oceans. And if you probably have heard about them, but one of the sort of things they're famous for doing, diatoms, is that they create, in special conditions, you get these big blooms forming. So they're sort of rather massive. So this is just off the coast here of the south of England, some blooms, and you might have seen these algal blooms. You can see them from space, basically. They're huge things. So they're a very, important part of the ocean, and they contribute about 50% of the global annual carbon dioxide fixation. So they're very important players in sort of biogeochemical cycling. And moreover, they're sort of right down here. If you look here at the bottom here, they form the basis for the marine food web. Yeah. So if things go horribly wrong with them, then there's going to be there could be potential consequences for the whole of the food web. So and basically they're important little critters. So what we've been interested in, or what Thomas has been sort of the focus of the work that he's been doing, when he came to UEA, that he started to explain to me, is the sort of effect of temperature on these organisms. And just to sort of emphasize, we know that by measurements alone at different depths, we know that the ocean temperatures are rising. Okay, so that's not really very controversial, and there are predictions, which might be a bit more controversial, that this warming will continue. Okay, so these oceans are going to probably continue warming up for whatever reason, and what we're interested in is how temperature drives the diversity of these phytoplankton. It's sort of predicted that it might reduce diversity in tropical regions, but we need real data to sort of start understanding whether that's true or not, and also to understand a little bit more about the relationship between temperature and the metabolism of phytoplankton in the environment. Okay, so so in this project that I got involved in about 3 years ago, we wanted to sort of the aim was to sort of use this new sorts of metatranscriptomics data to identify how temperature impacts metabolism and resource allocation globally, and to link the sort of structures, the algal physiology, to try and understand that better, to actually make some, eventually, hopefully, predictions on how that might affect biogeochemical cycling in the ocean. So those are actually quite, you can imagine, big aims, but we're sort of, I think we're already somewhere down the road there. So the approach we're taking is we're going to explore what's out there a bit using metatranscriptomics. Then in the labs, Thomas's group are actually going to do some very specific experiments to sort of confirm, at least for special examples, that these trends that we're finding actually appear to be correct. And then eventually, well, already we've started, sort of make some models and try and predict what might happen next. So here, I just thought it'd be nice. So we'll start with the kind of collecting part. So this is a picture of some people collecting some phytoplankton. One of the PhD students of Thomas Mock is Katrin, and Katrin Schmidt, and she sent me these pictures, which I think are rather nice, sort of give you a flavour of the kind of work that they're doing. So they go off in ships, actually go and collect some samples, and then here we are on the ship, actually with all these samples. So they actually go to various places in the world on a ship for a few weeks and actually go collect these things. And the data sets that we started with were sort of collected, I would say, by different groups in sort of a range of areas to just try to start getting a feeling. Okay, we have here Arctic, very, very cold, then slightly less so here. Then we've got off here the Pacific coast, down to the tropics, where we have much warmer temperatures, and also at the other pole. Okay, so these samples, we were basically given 5 data sets to analyze. As I said, I'm not a biologist, but if you're interested, you can probably understand this more carefully at the end. But essentially what happens, because I've put it into English here for me, is that we sort of the samples get size filtered so that we just try to get out the microbes, Then all the RNA is extracted. Then essentially we target the messenger RNA. We're not interested in non-coding RNAs, etc. Although I'm personally very interested in those kinds of things. That would be really cool to look at those. They're taken away. And then in this example, the samples were prepared for 454 sequencing, because back then, when these were done, that was kind of the method that was chosen. So here are some figures from the sequencing from the different areas, and you can see, well, we get rather large data sets. I think that's not really surprising after all the talks we've seen this week. One thing that's getting clearer and clearer is that these data sets are very large and they're getting larger all the time. And what we then did, or what Andrew did, and that was really part of his PhD, was to sort of create a pipeline to actually go through all of this and analyze and look at that data and try and understand. So first, there's some sort of quality checking, some clustering to remove redundancy. Then the taxonomy is done using Thimble, and that had the advantage that we could actually extend the database to include more marine eukaryotic genomes. And then we did some work, or he did some work on checking pathways, etc., which I'll just now briefly describe. So here we go. That's just a fairly standard, I guess, pipeline for trying to understand these kinds of data sets. I asked Andrew, though, I said, well, okay, well, we've had all these discussions about assembly or not assembly. He said that we did try assembly, but we got varying results. And for high diversity data sets, we got actually quite poor assemblies. And actually, because of these organisms, it's really difficult to assemble even a single species. They have really huge genomes, yeah, because we're looking not looking at the bacteria and stuff. We're looking at the eukaryotes here. So, it's not clear that assembly's that helpful here. But we're in this study we were mainly interested in counting sort of protein domains. So maybe it was not so important to assemble anyway. So I think assembly, maybe it really depends a lot on your question. And anyway, if you're a bioinformatician, you can either assemble or not. You can probably do both. I guess the problem will become when we get really, really huge, more and more huge data sets, we might then have to think seriously about whether we're going to try that or not. So here are some just basic figures of the various reads and where they were. And as you can see, most of the, or as I say here, most of the sequences were sort of ecosystem specific. So in other words, these kind of edges here they didn't intersect at all with any other ecosystem. So most of the stuff was sort of specific to where it was sampled. And indeed, as again, no probably big surprise after the talks we've been seeing, a very small percentage of the sequences actually hit PFAM. So roughly 70% of this stuff data was in no database. So the question is, well, what do we do with all this dark matter as we've been discussing? Well, obviously we can store it all, but or maybe as sequencing gets cheaper and cheaper, we won't care. We'll just keep the bits that interest us, because we can just keep going and do it cheaper and cheaper. But I think one thing we have seen this week is, especially with some of the talks about new techniques for sequencing, it would be very interesting to work out in future what we're going to do with all this data that we can't really map to anything. So yeah, in the taxonomic analysis, what happened was that we basically got a clear split. For polar samples, it was dominated by something called diatoms, and in the tropical temperate regions, there were alveolates. Okay, so there was a definite kind of, in terms of the different organisms present in the different sampling locations, we found that there was a clear split. So that's fairly good to see. That I would expect also that a lot of this might be actually confirming some things that people know. But what's nice is that in the actual metatranscriptomic data, we could actually find these things. And I think we've seen again lots of that kind of work this week already, where we're kind of using metatranscriptomes, etc., to confirm suspicions. We also did some multidimensional scaling just to see how the data, without telling it where to go, how it sort of fell. And again, it split quite nicely into different phyla, and at the same time it also split according to latitude. So we found, so the colder stuff was together, and the warmer, and then the Arctic, Antarctic here. There was, I was having a nice chat just a couple of days ago about whether go-term analysis is worth doing or not. Okay, I mean, we obviously there's a lot of problems with that, but we did do such an analysis, and we found that in the sort of hot areas, you get a lot more of the sort of processes that are getting flagged up are to do with things like photosynthesis, whereas in the colder areas, we're getting much more sort of stuff going on with translation. So patterns start to emerge. So it's about, I guess, because as Wally said, I was trained as a mathematician, I just start to find it interesting to look for patterns in the data. And again, if you do a sort of KEGG pathways analysis, you find that in these intricate webs of metabolism, that the photosynthesis pathways are dominating in the tropical and in the temperate region. So you get a lot of this stuff lighting up, whereas in the polar regions, much less of this is lighting up, and then nucleotide metabolism dominates. So again, a lot of what seems to be going on there in the actual oceans is to do with translation. And that was confirmed again, doing a more sort of careful analysis. We found that that things tended to cluster by environmental type, which is useful to know, and that translation was the most abundant across the samples, and there was a sort of gradient of translation according to temperature, and then sort of just checking that more carefully, just to do some correlation here, you find that you're getting much more sort of translation going on in colder, and then less so in the warmer regions. So I mean, unless you're really interested in phytoplankton, and I suppose quite a few of you here probably aren't, I think the main thing that I find interesting is that we can actually really start to dig out patterns again from this data by cutting it up in different ways. So in your talk yesterday, I was fascinated with this morphological transformation where maybe we can start to see even groups of things that move together. So I think we're at early days probably with all these big data sets, but it does seem they contain all sorts of interesting information, and maybe we need more sorts of data mining approaches as well to not, I mean, this is hypothesis driven research in a sense, because Thomas and his group have been interested in the way temperature affects things, but we've got lots of data sets here. There are probably things hidden in there that we aren't looking for that other people might be able to dig out and find interesting too. Whoops. Gone to sleep or what? Oh, that's not very nice. Seems that it's crashed. That's unusual. Alright, now what's the date today? 27th, is it? Yeah, it's just seems that the whole thing just crashed. So that's quite strange. Yeah. No, I've got here. That's not what I want. What's this? Hopefully, this is the right one. It's strange that it isn't even appearing on there. Wait, I'll just get it to where I was. Okay, that's where I was at. Now, why is it not? Ah, there it is. Strange.
Maybe you just
Did I? Ah, I might
Yeah.
have pressed one of those.
Yeah.
Oh, they should move those buttons somewhere else.
Yeah.
Get a new engineer on the... Okay, what's this? Logitech, okay. Yeah, so as I said, I mean, it was obviously these kinds of predictions that you, or these patterns that you see in nature, it's important to sort of check that we're really seeing them in the lab too, to make sure that we're not, in a sense, it's always nice to tell a story, isn't it, from data, but then I think in the end you've got the burden of proof is to actually check carefully that the kinds of trends that you're finding really happen. Now, of course, you can't take all of the different phytoplankton in the ocean and grow them all up in a lab. We've heard that many things don't grow in the lab, and of course it would just be prohibitive, but at least Thomas's group were able to check for a couple of these diatoms that we really did see the kinds of trends that if it was colder there was more translation. And they also did some experiments which showed that at higher temperatures the translational efficiency was actually higher. So that's all very interesting, I think. Whoops, what's going on now? So yeah, so in summary, from the sort of data analysis of what we've looked at, we found that there's sort of a correlation between temperature and translation in naturally occurring communities based on the metatranscriptome, and that's also true for model species under laboratory conditions. So what's that all mean though? I mean, okay, that's an interesting story in itself, I think, but what kind of implications could that have? So if you have more ribosomes, okay, then you get more rRNA, and that's the largest pool of cellular phosphorus apparently. And so that means, so in here we have, so for example, so in cold temperature here we have lots and lots more rRNA, and in warm we have less. So this can affect, for example, the MP ratio. I mean, of course you could pick probably all sorts of ratios if you chose, but at least this particular temperature effect will have some effect on the MP ratio. So what could we actually maybe start to do some modelling of what that might imply? So as I mentioned, Tim Lenton and his colleagues have been developing now for several years some models for growth and resource allocation, and actually in phytoplankton. So I don't know all of the details, that's why I feel a little out of my comfort zone, but I have done a little bit of modelling, and you can imagine that you can set up sort of cellular models and put all of your different parameters, metabolism, etc. in there. Just depends on what kind of granularity you're interested in as to how detailed that model becomes. But they have a model, and what I think was really fascinating, I mean, you can of course just make a model of a single cell, but what they do is actually now use this as a sort of building block for an agent-based model of a marine ecosystem that they then feed into a general circulation model for the ocean. So in this general circulation model, you have you sort of cut up the globe into little grids, squares, and you have all sorts of, when we actually do all these climate predictions and weather predictions and stuff, that kind of mathematics, they can feed now their models of these cells. They put little agents in sort of the little grids, and they all interact with each other and grab phosphorus or etc. And you can sort of then use that to make some kinds of predictions. Okay. And so for the MP ratio, which we concentrated on here, they found that if you actually raise you can do that in it's possibly going to happen anyway, that things are going to go up 5 degrees. But if they go up 5 degrees, they were finding that we were getting a large MP ratio increase here, which I was, when I first came across this, I thought, okay, that's interesting, but so what? Well, then apparently that could lead to nitrogen limitation, and then that could have potentially unpleasant consequences for the food web. So I think rather than try and describe all of that model in detail, which isn't mine anyway, I think what I kind of why I wanted to bring that to your attention is I think it's rather nice that we can start now to take our data and put it into such models. I've seen in the first talk of the conference, we also kind of got the idea that using metabolic models, we can actually maybe start to inform our models and maybe go beyond, and actually just simply sampling and seeing what's there, which is an important thing, but we can actually maybe start to use that to do some modeling. And I think that probably is very relevant to the next, program that's going to be running later this year. And indeed, I've spent some time at one point modeling denitrification, and I can imagine in chemostats and such like, you could again build models with agent-based models. I know people do these things, and then maybe we can start bridging the gap between pure data collection and analysis and modeling. Yeah, I mean, again, for things like, maybe that's already being done with these biogas. So this was almost a sort of, although it seems to have taken quite a long time, it was really a beginning study for a more in-depth study that's now going ahead. So Catherine has been on the ship, and they're collecting a lot more samples across different temperature zones, yeah, and JGI and EBI. Well, JGI is going to do a lot of the sequencing of all of this, and we're going to get metatranscriptome, metagenome, etc. data. So, and we're already getting terabytes of this data in a pilot study. So we're going to get a lot more depth of data, which sometimes makes my palms start sweating, because I think, okay, we're going to get all this terabytes of stuff, and I think, like everyone in the room, we're getting to a point where, if we're not careful, we will have a bottleneck of, who's going to do all this analysis. I mean, but most bioinformaticians that I know have been saying that for years. So, in some sense, maybe that's just always going to be the case, or maybe it's got to come a point in the future where people have to really pump a lot more money into training and bioinformaticians to actually, so that we can actually start doing something with all this data. And there's like all sorts of nasty, so in terms of problems, bioinformatic problems, there's all sorts of nasty problems that we have to deal with when we're looking at this eukaryotic data. I mean, many of the, for obvious reasons, many of the tools have been set up to deal with prokaryotic work, but we're going to have to deal with all this eukaryotic data, and assembly is going to be pretty unpleasant. We've got very few reference data sets, and we're going to get truckloads of data. I mean, that's exciting too, yeah? I mean, and actually, in terms of reference data sets, just to bring this to your attention, there's the marine microbial eukaryotic transcriptome sequencing project is now going ahead, and they're aiming to sequence the transcriptomes of about 750 samples from hundreds of diverse organisms. So we're going to have lots of very interesting data. And now in terms of the sort of things like pathway analysis, which we've done at sort of an exploratory level at the moment, I wonder, is there, how much confidence can we have in this? We've had discussions about Go terms and stuff, and okay, these pictures, I think they are actually quite helpful. I mean, to visualize what's going on in the data, but can we what kinds of level of conclusions can we draw from this kind of work, and how could we maybe start getting towards using this to actually inform our models, yeah? In a more sensible way. So yeah, that's really all I wanted to say today. I'm going to just say, if anyone knows a student who wants to do a PhD in this area, then please let me know, because we have a studentship and I'd love to hear from them. So thanks very much. Yeah. Oh, sorry. I should let Wally.
The modeling that you're doing, where you're looking at the effect of temperature and you're looking at nitrogen and phosphate limitation, is really interesting. But I'm wondering if there may also be a danger when we do the modeling that there are external pieces of information that are still missing, like cardinal temperature.
Yeah.
What happens, when we have a maximum temperature and you exceed that, and where there's predation with the diatoms and such. And I'm just wondering.
I mean, absolutely, for sure. Yeah, there's going to be a lot of things that we're not including in these models. I mean, and as with most mathematical models, we normally start fairly. I mean, these aren't, as far as I can see, because I don't develop these models, but these already look quite complicated models. But I agree, eventually you might want to include more factors. But then we always have that problem that if we allow ourselves too many parameters, we might end up with a model that could basically predict anything, just depends on what the parameters are. So I think the challenge is going to be, as with most of these kinds of mathematical models, is to get good estimates, robust, sensible estimates of parameters, and then maybe work hard at deciding what is and what isn't important as well, which, I mean, that's a standard, I would think, problem in mathematical modeling, but I agree. It would indeed, these things are somewhat focused on a particular problem, and if you wanted to do them more generally, yeah, you would certainly have to include probably more factors, yeah.
So you've shown some correlation between temperature and the growth, but is it also important to understand what's the cause and effect actually there? Because perhaps as the growth going on, do they also have an effect on temperature? And then if you would include that in the model, what causes what actually? So how does your model, does the prediction?
Well, I can't speak about the model itself, but I agree. I mean, obviously, what's cause and effect, you have to work out what is what, yeah? But, but what exactly was your question there? So,
no, I mean, so when you model something like that, right, I wonder if it is really important to know what is happening. So usually, if one parameter is quite dependent on another one,
yeah, indeed, yeah. I would expect as with most modelling, yeah, you're going to do some sort of parameter analysis to see which parameters are most important and which are not, and maybe which are correlated or not. I don't know what they've done in that direction though, but I mean, I can talk with you afterwards and put you in touch with the modellers if you like.
Yeah, at the very beginning you showed that Venn diagram where there was much overlap between the different data sets, and then you told us that the genomes are up to 100 gigabases in size, so I'm not sure how big the transcriptome would be, but could that just be that the hundreds of thousands of reads that you sequenced were just not enough?
Yeah, that could be, yeah. It will be interesting to see as we get to sequence more and more whether those will start to overlap. Yeah, no, I agree, that's a good point. It's not clear at all whether that might be the effect, that we just haven't basically sampled enough, yeah?
I had a comment on that line myself. I mean, if you want to include more factors, which you would need to do when you want to get down to modelling these systems, you're going to need a lot more samples with all kinds of levels of factors in there. So that's going to create, interestingly, the study you mentioned about the ship going down the coast of Africa, you know, collecting lots of samples, I imagine. You're going to get vast data sets in the future, and it's kind of worrying to think that there's nobody really going to have the time to analyze it all.
Well, didn't Volker say we should send? Volker said we should send all data to
Well,
MGRAST. So
I mean,
Okay.
I mean, the truth of the matter is that this, the way it's going now, is that, you know, in 10 years' time, we might well be sitting here again saying, we've got all this, we just are swimming and dying in data, yeah? It's just deluged in data. I mean, and the big question will be, in my mind, is how, I mean, we've already heard that Richard Durbin already said, you know, in his opinion, this is going to be a huge problem. I mean, and that's got to be something that we've got to do in terms of infrastructure, actual people trained to actually use the infrastructure. I mean, I just see that as, I mean, again and again we've heard it's probably the limiting factor, yeah? I mean, now as for how that will inform models, well, I mean, I could imagine this kind of work going on with things like chemostats, yeah? Where you might have a little bit more control on the environment, okay? So you could have, and again, these, maybe even these biogas reactors, but they're probably already horribly complicated. But it would be nice to see, or maybe with those examples where we saw with the little test tubes of soil, where we could actually try to identify what's important, what's not, so that we can make models. But we already saw models on, was it yesterday, where they were predicting about the sulfur, how that was, the sulfur cycle. I don't know, I mean, modeling is a funny thing, yeah, because when you start to set out to model things, often you quite quickly have to focus in on one specific piece of your problem, because it's just too complicated otherwise. And then your models get very simple, and so is the fact that they're simple really a bad thing? Or should we just, I think most of the time we carry on and we hope that the models will inform what we should do next, at least, even if they're not accurate.
Further questions? I think with that would thank Vincent very much for that very interesting.